My name is Alyssa Atley. I'm GlycoNet's Communications Director. Welcome to our first webinar about GlycoNet and its integrated services. I will pass off the webinar to our speaker today, Dr. Warren Wakarchuk, who is GlycoNet's Scientific Director and a professor at the University of Alberta.
All right. Thanks, Alyssa. And thanks everybody for taking time out of what I know is a busy time of year, especially for those of us in academia, to come in and hear about GlycoNet and in particular GlycoNet integrated services. So I would like to start by giving a land acknowledgement, and many of you have probably seen land acknowledgements before, but I tend to do them slightly differently, and I wanted to take a minute just to think about why we do a land acknowledgement. And so in case you don't know, these are done in support of a process called Truth and Reconciliation with the Indigenous people of Canada who had suffered horrific treatment by the Canadian government through what was known as the residential school system. And many of us in academia give land acknowledgements. And the idea here is that it's supposed to provide some time for you to think about the plight of Indigenous people in Canada, and in particular, thinking about what happened in the past and what changes we can make going forward. And in order to do that, you have to know a little bit about the Indigenous land that you're on. And so I put the map in the left-hand corner of the screen here is one that shows various regions of Canada and various treaties and agreements that were made with Indigenous people. And normally when we give these land acknowledgements, we talk a little bit about the people whose land we are on. And today I don't want to take too much time because, as you're going to see, GlycoNet integrated services is spread across the country, but I will mention that in Montreal, the land is on unceded, or the land is unceded territory, which meant that the Indigenous never gave the land up in a treaty. And in Hamilton, it's the Dish with One Spoon Wampum Belt Covenant. And in Toronto, it's also the Dish with One Spoon, but there's also a Treaty 13 with the Mississaugas of the Credit River. And in Edmonton, it's Treaty 6, 7, and 8, and the Métis homeland. And in Vancouver or Burnaby, again, this is unceded territory. All right, so let's talk a little bit about Glycomics. So why do we care about Glycomics? I presume that everybody who's here either is hoping to learn more about Glycomics or is somehow involved. And I like to give a little 30-second biochemistry lecture on what is Glycomics and why do we need to have integrated services around them. And it's because glycans coat pretty much every cell. And because of that, then they are responsible for the molecular recognition in much of biology. And the schematic here, I should mention, was designed by a former graduate student here at Saxena, and it depicts a bunch of different interactions that can happen that are mediated by protein-glycan interaction. And so pathogens, obviously, antibodies, toxins, soluble lectins, things like galectins, which are circulating in the blood. And then, of course, cell receptors recognize glycans all the time. And what this means is that they are intimately involved in health and disease. And more importantly, this information is not encoded in the DNA. And so in order to study it, you can't simply look at what genes are present, or even when the genes are there, when they turn on and off, because there's a whole post-translational part of how glycans end up where they end up. So it's a little bit more specialized than simply sequencing or determining the protein content. So GlycoNet is a pan-Canadian organization, research organization. It started off as being funded through something called the National Centers of Excellence program, and it started in 2015. And we recently transitioned from being funded through the NCE program, which stopped, to being funded through something called the Strategic Science Fund, which we just started in 2024. And the year before, we were funded through something called the Canadian Foundation for Innovation Major Science Initiative. And this is money that was given directly to support the GlycoNet integrated services, which all started as part of the NCE. So it was kind of baked in to the way that the NCE was set up. We needed analytical facilities to facilitate the research that GlycoNet was doing. And so what that meant is that we had facilities to help us with our mission, which is to conduct and translate glycomics research into real-world applications with an eye toward commercializing these technologies and obviously create some kind of socioeconomic value. So I mentioned that GlycoNet Integrated Services is a distributed facility. So we have 7 different locations across the country, and they're depicted on this map. And as I mentioned, the funding started in 2023, and importantly, it is leveraging over $35 million worth of CFI-funded infrastructure that is present at these locations. And so this is a nice way of making sure that those facilities are continuing to be used at something near capacity in order to do what I think is fantastic support for the glycomics community here in Canada and abroad. The 7 locations hosts 14 different nodes, if you want to think of it that way. So 14 different activities, different labs. And if we start on the right-hand side, so the easternmost part of Canada, and we look at what's happening in the Quebec region. So we have a group at the Institut Armand Frappier in Laval, which is a suburb of Montreal, where we have glycan analysis and molecular glycobiology, and I'll give more details about this as we go through the presentation today. At McGill, we have glycan analysis, mainly centered around doing microbial glycans, and in particular, a lot of really excellent work on fungal glycans there. We have a node at the hospital at Saint-Justine called the CHU. And again, this is glycan, very specialized glycan analysis, mostly in support of clinical work. And we'll talk about that. And then in Hamilton, we have, in Toronto, we have Drug Discovery and High Throughput Screening Center, which is called Spark. And this is housed in the Hospital for Sick Children in Toronto. And then we have another drug discovery and high throughput screening facility in McMaster in McMaster. And they're mainly looking at infectious disease related research, but not exclusively. And then if we skip the big target in the middle of the country for a minute and go all the way over to the West Coast, at Simon Fraser University, there's also the 3rd drug discovery and high throughput facility, which is housed at, as I said, Simon Fraser. GlycoNet's administrative offices are housed here in Edmonton, where I'm speaking from today. And we also have a high concentration of nodes that do a bunch of different things. So we have carbohydrate synthesis here, we have glycan analysis, we have glycan ligand screening and discovery, we have glycoengineering and molecular glycobiology, all of which I will talk a little bit more in the following slides. So when we were thinking about what is it that we actually do? I think we just decided that we should put kind of a pipeline schematic together to show people what in fact we're doing. And so if we start on the left-hand side of the slide here, with the glycan of interest. Obviously, determining the glycan structure is important. We can then define the proteins that they interact with and the kinds of interactions that they have. This can then lead to some kind of bioassay development. And this is where the high throughput screening facilities come in. And then this allows you to screen for some kind of glycan mimic in a library, which can then lead in a couple of different directions. So this can lead to then chemical synthesis for conversion of a compound into a biological probe to look more closely at the biochemistry that's going on there. Or you'll see on the grayed out side, it can lead to some kind of medicinal chemistry campaign to convert the hits into drug candidates. And I've grayed out the medicinal chemistry and the conversion into drug candidates because it's not something that is actively happening in the GlycoNet Integrated Services laboratories. It is happening in the labs of collaborators and other partners, but it's not actually part of the funded part of GIS. Going back to the left-hand side, you could also take that glycan of interest, perform some chemical modifications to turn this into a biological probe, which would allow you to do some biophysical characterization of the molecules that interact with the glycans. And you can see how all of these things then sort of feed together, all starting off with knowing something about the glycan of interest. So let me start with talking about the high throughput screening facilities. I think we're very fortunate. Canada is not a very big population base, but in terms of scientific cooperation, I think we're pretty good at it. And I think GlycoNet and GlycoNet Integrated Services are an even better example of how that can work. And so these three screening centers have been front and center in a lot of the work that has come through the GlycoNet funding programs. And now, of course, part of the services that we offer to the broader scientific community. And what's really interesting about these three screening facilities is that really they are overlapping, but have unique capabilities at the same time. And so it's really a very interesting and vibrant group of people doing some very cool things. I've been fortunate to visit all three of these facilities, and it really does look like something out of a sci-fi movie when you go there. The amount of robotic stuff that's happening, and just the idea that you can screen through hundreds of thousands of compounds looking for things in a variety of assays. And I think that's the strength of these three combined facilities, is that we've got things like cell-based screening, a lot of image-based screening, so high content imaging, which is really important. This is either done from model organisms or things like zebrafish, for example, or from live tissue culture cells. At the Spark Center, they've been looking a lot at working with organoids. And you can see in the middle of this slide, there's some overlap in terms of screening, looking at enzyme inhibitors, for example, image-based screening, live cells, fixed cells, stem cell derived cells. The McMaster facility, very good with complex bacterial assays, and some really, really interesting ways to probe biology, in ways that are, like I said, pretty sci-fi-esque, but leading to some really cool science coming out the other end. So in terms of Team HTS, on the left coast, we've got the Center for High Throughput Chemical Biology, which is co-directed by Roger Linnington and David Vocadlo. David, probably well known to the glyco community, and they are ably assisted by Sandra, who's helping out. And I would have to say that when I look at the kinds of things that they can do, it's pretty spectacular. They've had a lot of interaction with company sponsored work. Um, and if people are interested in more about what they do, the email contact for the folks are all given here. As I mentioned, the Center for Microbial Chemical Biology at McMaster, the principal investigator there is Eric Brown. But a lot of what's happening there these days is actually headed up by Tracy Campbell, who's the manager of that facility. And then if we move to the Spark Drug Discovery Center at the Hospital for Sick Kids, this again is a co-director scenario where we have Roman Melnick and Daniela Roten, who are the co-directors, and they are ably assisted by Chris Flad, who is the longtime manager of the facility. So I thought it would be interesting to just put up a couple of sort of challenges and opportunities to highlight what the high throughput screening centers are doing. And as I mentioned, I took a chunk out of our pipeline analogy on the left-hand slide here. So really, bioassay development is really important for high throughput screening. And when I visited the Spark Center at Toronto, I was talking to Chris Flad, and they spend quite a bit of time just helping clients with that bioassay development, because a lot of times you don't have a good idea about how to do that. And I think that's a typical scenario for these high throughput screening facilities, in that people will need help to design a proper bioassay in order to find hits for the things that they are interested in. So in the biomedical space, patient cell-based assays are really what are important for efficient screening, because the standard cancer cell lines most certainly are not representative of anything that would be useful for screening for a drug candidate. And they've been used in the past, but I think nowadays it's fairly straightforward to actually get patient derived cells. And so GIS has many of these protocols in place and has really been at the forefront of developing molecules for cell-based assays in Glycomics. One of the other challenges, of course, is that specialized chemical probes and substrates are required for this screening. And one of the things I would have to say that HCTB at SFU is very good at is looking at those sort of reporter molecules, some very cool fluorescent reporter molecules. And I would say that really GIS has some world-leading expertise in the chemical biology space for glycomics applications. One of the things that I thought was really interesting, and that I didn't know a whole lot about until I started talking to the folks at the screening facilities, is that screening of natural products obviously has been around for a long time, but GIS actually has access to probably the largest compound collection in Canada, and that there's several varieties of these natural product libraries, which makes it very interesting to do screens, of course. And in the context of looking for new antibiotics, which is something that the center in Hamilton has been doing, this, of course, is very important. So I want to give you a couple of examples, and I wanted to preface talking about these examples in that it wasn't possible to show a lot of examples of client-based work, simply because in many cases the clients would like to maintain some confidentiality about what was happening. So if these things look very generic to you, there's a reason for that. But I think it illustrates the point. So in this particular case, this is a screen that came from the Center for High Throughput Chemical Biology at SFU. And it illustrates an enzyme assay-based approach using cells, not purified enzyme. And it's a 5-minute incubation with the compound library and a 20-minute incubation with the fluorescent reporter. And they were able to screen 80,000 compounds in 3 days. And that's a phenomenal number of compounds to go through. And when you've got a very nice assay. And for those of you who aren't familiar with what HTS data looks like when it comes out the other end, in this case, looking for an enzyme inhibitor. So looking at percent inhibition, and obviously at the 100% line here, there are some positive controls here that are 100% inhibiting things, and then a scatter of things. And the way that hits are determined is by how many standard deviations there are from some cutoff. And in this particular case, there were 800 hits that met this criteria out of 80,000 compounds. And there were around 400 that had a 35% inhibition cutoff. And that's kind of starting place for paring down this 400 hits. And I should mention that this particular screen is for an enzyme target that is important in CNS diseases. A second example that I got from Roman Melnick from Spark is one, it's a high throughput screen looking at some emerging pathogen targets. And in this particular case, collaborative effort with Dr. Jennifer Geddes McAllister at the University of Guelph. And in this particular case, 2 hospital-acquired pathogens, Klebsiella pneumonia and Cryptococcus neoformans, well known in the hospital infection area, as well as attaching themselves to immunocompromised patients. And both of these organisms have a capsular polysaccharide that's associated with disease. And in this particular example, what is shown is the data from a single 384 well plate. And this is a very simple assay. I like the simplicity of this assay. It's a growth, no growth assay of one of the organisms. And Roman didn't tell me which organism this was. I'm guessing it's probably the bacterial one. But you can see that there's kind of a band where there's some minimal effects. And then there's one single compound here, one spot corresponds to a compound where there's some nice knockdown of the growth. And so this is the way that this kind of assay can be used. And as I said, I like the simplicity of this one. This one's growth, no growth. That's relatively easy to score. So nice example. At the McMaster facility and the other two, I have to say, the question is really what can we do for you? And the idea is that the staff at the centers there are trying to facilitate your research. So I'm pretty sure that everybody can obtain some training for the users. Consultation and experimental design assistance is really critical for these. And as I mentioned, if you don't have a good handle on what your bioassay is, you can talk to the experts and they can help you design, design what kind of an experiment you're going to do. And in many cases, there is staff available to help you complete the experiments if you need extra assistance, or if you're comfortable and want the facility to do the experiment for you, that could certainly be arranged as well. And you'll notice that I've put contact information here, just in case people are interested in getting a hold of any of the managers or any of the PIs from these facilities. And it could be that if the center that you contact can't help you, maybe one of the other two could do the job. All right, so let's move on and talk about Team Quebec for a minute. So we have 4, as I mentioned, 4 locations So we've got, well, actually 3 locations for nodes. We have analytical R&D at Saint-Justine. We've got the biophysical and structural biology analysis at INRS, as well as molecular glycobiology at INRS, and microbial glycome analysis at the McGill Center. So at Saint-Justine, they were able to secure a really very space-age instrument, which is from the Water Select series. When you see this thing in real life, it doesn't look like a scientific instrument because it's black and shiny. It doesn't have tubes and wires running everywhere, at least on the outside. But it's a very, very cool instrument in that it's so versatile. It does a variety of things. It does standard LC-MS. It'll help you do things like metabolomics, for example, but it's also good for imaging. And we're going to talk a little bit more about imaging mass spectrometry when we get to the Alberta node. So this is a center, as I mentioned, that is ensconced in a hospital. So mostly doing clinical samples, but they have some really nice expertise there in order to facilitate things like targeted or untargeted metabolomics in that clinical setting, or looking at particular glycan metabolites. And I know that there's a special interest there in glycolipids and glycosaminoglycans, which aren't necessarily that easy to study. If we move to the Research Institute at the McGill University Hospital Center, so this is the Microbial Glycomics Center. And if we just look at the left-hand side of the slide here for a minute, Francois Lemouf, who is the manager of this facility, has been instrumental in helping to analyze a series of enzymes that work on mostly degrading polysaccharides. And he also simultaneously was working on the polysaccharides themselves to try to figure out, what they were composed of, so that they could figure out, what the substrates for these enzymes were. He's put a lot of effort into the determination of protein carbohydrate ligands, and specifically looking at fungal cell wall glycans, which are not trivial to analyze. And lately, he's been doing some really cool stuff with peptidoglycan from various pathogens, things like Mycobacterium tuberculosis, for example. And it turns out you can get some very nice signature mass specs, which are shown on the right-hand side of the slide here. But the kind of work that they can do is not limited to peptidoglycan. They can do other cell wall components, capsules, extracellular vesicle polysaccharides, and the like. And Francois supplied me with a QR code in case people want more information and want to get a hold of him. At the INRS, the Institut Armand Frappier, there's a very long list of things that this facility can do. I'm not going to read through them all because I'm sure you can read them faster than I can. But a lot of these pertain to biophysical characterization to define protein interactions with particular molecules. A couple of things that we're kind of keeping our eye on are some assays that are coming up for the maple syrup industry. I don't know if people know this, but the maple syrup industry in Quebec is worth about $700 million to the Canadian economy. And when we ship maple syrup abroad, people like to adulterate it and pass it off as pure maple syrup. And so what they're thinking of is some kind of quality assurance, which is going to be based on the sugar content of the maple syrup as determined by NMR. So there's some cool, very Canadian things coming out of this particular group. So let's move on to the University of Alberta here. So here are some of the nodes. So we have glycan ligand screening headed up by John Klassen, who really is one of the pioneers in native mass spectrometry, looking at proteins binding ligands in the gas phase. It's kind of cool stuff. But he's also now very interested in imaging mass spectrometry, and I'm going to talk about that in a minute. And then underneath the glycan ligand screening, we have the molecular glycobiology unit #1, headed up by Matt McCauley. This is some very sophisticated work engineering mice and cell lines in support of glycomics research. And on the right-hand slide, we have the glycan ligand discovery groups. So there's two different groups here. So one headed up by Ratmir Durda. And really, this is a very interesting technology using a phage display technique that I'll explain in a little more detail in a minute. And then underneath Ratmir is Lara Mahal. And Lara Mahal's lab has been instrumental in exploiting something called a lectin microarray. And I'll mention more about the lectin microarray on the next slide, but it's a really nice high throughput method for looking at biological samples. So as I said, this lectin microarray, basically what you can do is take tissue, things like serum, cell culture, you can extract the glycoproteins, you tag them, and then you've got a reference versus your screening sample. You take those glycoproteins and then put them onto an array that has carbohydrate binding proteins on it. And so this is kind of the inverse, if you like, of a carbohydrate microarray where the carbohydrate is on the slide and you're probing for binding proteins. This is the other way around, where the binding proteins are arrayed and you're looking for binding of a particular glycan structure. And these are then scanned, looking for the fluorescent signal. And you end up with these really complicated heat maps, which take some time to wrap your head around. But there's a lot of data in here. And the number of lectins that are on this array really do help discriminate what the actual pendant groups at the end of these glycans are. And it really helps you narrow down what you want to do next. And so it is possible to do hundreds of samples this way. It requires small amounts of protein, and it's a really nice way of identifying probes for further work. And then, of course, the epitope would need to be further refined by other analysis. But when you have hundreds of samples that you want to process, this is probably how you want to start it. All right, the liquid glycan array. So this is the work that's being done in Ratmir Durda's group. And this is really fits under the heading of, if you want to go back to our pipeline definition of converting things into biological probes to define protein interactions. And this is a very sophisticated assay where what's happening is you have a filamentous E. coli phage, which has got a DNA barcode inserted into the non-coding region of the genome. And what that allows you to do then is to have different barcodes for different phages, and then these phages can be chemically modified to carry different glycans. And what's interesting about that is that you can then mix them all together, and you can stick them into a mouse, you can use them on tissue culture, you can look at purified proteins, and you can identify the binders by using next-gen sequencing, specifically looking for the barcode. And this is really kind of a nice technique if you want to do this in vivo, because now you've got some, you can control the density of the glycans, you can control the complexity of the glycans, and you can find things really easily with this next-gen sequencing, right? The combination of PCR and next-gen sequencing to look for those barcodes. So it's a very sensitive technique, and I think it has a lot of promise for especially this in vivo work. All right, let's come back to imaging technology for a minute. So the kind of work that is being done, the one that I know the most about is the one here at the University of Alberta in John Classen's lab, where again, we're taking glycan of interest and we're trying to find glycan structures here. And the way that this is done is to prepare tissues. These could be fresh frozen, or they could be paraffin embedded. And you can put these onto a slide. You can get rid of if it's been embedded in a preservative, you can get rid of the preservative. You can treat with an enzyme to release the glycans. You can put a MALDI matrix onto the sample, and then you can do MALDI-MS imaging. And what these pictures show then is a heat map looking for N-link glycans. And blue is low signal and red is high signal. So you can very quickly see in a particular tissue slice, how dense a particular glycan is in a particular region. It's a very, very powerful technique. And John is working on some nice tricks to be able to distinguish different sialic acid linkages, for example, which of course is also important. And they've done some nice work looking at glycolipid targets. And this is a mouse brain slice shown on the bottom here, looking for the ganglioside GM2. And I should mention that this is all done on this Bruker 15 Tesla instrument, so gigantic magnet. And this one doesn't look as fancy as the one at Saint Justine, but it generates very similar data. It's a couple of years old now, but still working just fine. And again, as I mentioned, we have 3 centers that can do imaging mass spectrometry. So, but like I said, I know more about the one from here in U of A. So in terms of molecular glycobiology, what Matt's node has been able to do is to become very good at creating cell-based models using standard CRISPR-Cas9 or lentiviral gene delivery. They've been working on making transgenic mice and have all of the instrumentation to support that. And obviously, making mice takes a long time. So these kind of thing doesn't happen overnight, but they've had lots of interest in different cell lines and generating different mice for different projects. All right, so my own group, the one that I'm responsible for here, is what we call the GlycoNet Core Services Team, and it's called the Core Services Team because it used to be called Core Services when it was the NCE, and we looked after the analytical R&D as well as the carbohydrate synthesis, and we've been doing a lot of work looking at recombinant enzymes for glycoengineering. And so the team is shown here, and I've got a red box around Sichin Lin because Sichin is the node manager for the carbohydrate synthesis and for the analytical R&D. So what can we do in terms of carbohydrate synthesis? Well, we've got a very interesting automated glycan synthesizer, which is very good for making biological probes. And so this is part of our pipeline again, where we've got a glycan of interest and we use chemical synthesis. But in this case, we convert it into a biological probe. And the way this instrument works is that there's a bead with a chemical linker, and your sugar goes on the end here. And when it's released, it has this pentyl side chain with an amine group at the end, which you can conjugate to proteins. You can put on a fluorophore, you can put on a biotin, you can do whatever you like. So it's a very convenient way of being able to synthesize sort of, 10 to 20 milligram quantities of things that are going to be used for biological probes. In terms of the glycan analysis, we've been looking at releasing glycans, tagging them with a fluorophore, and then basically doing LCMS analysis. And this is what a client would see when they ask us for this analysis. So here are the HPLC traces from the different samples, and then from the mass analysis, the potential structure is given down here. And this is pretty straightforward these days. I will mention that we like to use the Waters kit just because that's a very nice fluorophore. It's sort of industry standard. A lot of pharma use that particular methodology. So it's very easy for us to do and gives quantitative information about the glycan that we're releasing. And I should mention, we can do this for both N and O glycans. The O glycan's a chemical release, but gives you similar looking data. I wanted to spend a couple of seconds talking about molecular glycobiology, and in this case, recombinant glycoengineering of biologicals. So we've been asked to do this a few times, and in this particular case, we're looking at remodeling N-glycans from a plant-derived therapeutic protein. And so we've had recombinant plant-produced proteins, which typically have this truncated N-glycan, which we then add back galactose and sialic acid. And the way that that actually looks when we look at an HPLC trace is the black is untreated, pink is when it's treated with the galactosyl transferase, and then the blue is what it looks like when it's treated with a silyl And so we're converting these truncated ones into the proper ones. And we're very good at doing that. We have a spectrum of enzymes that allow us to make different sialic acid linkages, for example. And we can do this on both N and O glycans. So in terms of new services that are coming along, being an enzymologist myself, we've been very interested in increasing the suite of enzymes that we have for wider polysaccharide analysis, and in particular, looking at heparin and chondroitin. So degrading them to look at compositional disaccharide analysis, which seems to be what most people would be interested in. We've got some enzymes for looking at some plant polysaccharides. We're still playing with those, trying to figure out what's the best way to be able to do an analysis from plant-based samples. We're really looking at improving N-glycan release, and in this case, we've had people ask about plant or insect proteins, and of course, plant and insect proteins have a fucose in the wrong spot, so PNGase F doesn't work. You need a different version of PNGase in order to make that work. So we've got that enzyme in testing. We're looking at a technique for getting N and O glycans from the same sample. So, and in particular, looking at things like tissue and cell lines. It's relatively straightforward to do it from a purified protein. It's a little bit more of a challenge to do it from cells. We've got a nice methodology for looking at ceramide-based glycolipid head groups with an enzymatic removal and then chemical tagging followed by HPLC, much like we were doing for the N and O glycans. And then we have a bunch of glycan sequencing and processing enzymes for taking off monosaccharides one at a time to help resolve some ambiguities. And as I mentioned, imaging multi-mass spec at 3 centers, so here at the U of A, also available at McGill, and then obviously at Saint Justin with the fancy space age instrument. And then glycoproteomics is coming along at McGill. And when I say glycoproteomics, what do I mean? I mean, you take a protein which has a bunch of N-glycan sites, and you are able to see the full spectrum of glycans that are present at each site using a peptide-based analysis. This is kind of an extreme example. This is a protein with lots of N-glycan sites, but the technology is working, and this is being done at McGill. So Francois Lemouffe is looking at getting that. It's already working. It's still needs some optimization, but if you have projects for which you need some glycopeptide analysis, we are able to offer that service now. And then, as I mentioned, our single sample dual glycan analysis is based on the idea that we have a methodology where we can immobilize things on PVDF surfaces. We can use GAG degrading enzymes to get rid of the GAGs that come from most tissue culture cells or tissues. And then we can do the enzymatic release of N-glycans before doing the O-glycan release. And then that way we can get both N and O. And we've tried this on a few cell lines. It works pretty well. So again, if people have interest in looking at that from cell lines, it's something that we're ready to offer as a service. All right, so where do you find us? If you go to the GlycoNet website, there will be a drop-down menu for services. You can click on that and you can get to the Integrated Services tab, and you can work your way through the Integrated Services tab to find the particular service that you want. There's a table. And then if the technique that you're interested in is not on the page, there is a request form, and there is a contact us where you put in your name, email, and comments or messages. And then we'll do our best to try to get back to people to see if we can help. And so with that, I'll end here. And if people have questions for me, I'd be happy to answer them.
You can also put a question in the chat if you have a question there.
Maybe give a shout out to Michelle and Mary France from NRC. Nice to see you guys here. Thank you for coming.
Maybe while we're waiting for any questions, I'll just also mention a link to the recording of the webinar will be sent via email afterwards. Also, we are organizing more webinars that go into a bit more detail about different areas of services. And our next webinar will be on March 5th, covering glycan analysis and synthesis. If you would like to sign up for emails, if you didn't select that when you signed up, just send us an email and we'd be happy to add you to the list. Or you can also follow us on social media where we share our upcoming events and opportunities as well.
Yeah, and maybe just to add, if you're not sure who to contact, I mean, you can email me, you can email pretty much anybody from the GlycoNet team in order to help, direct you to a service that that you might need. So, yeah.